This is chapter 12, part 10. In this video, we are going to be discussing synapses. Remember when we talked about muscles and neuromuscular junctions, that a synapse is where communication occurs. So in this chapter, we're going to talk about communication between two neurons. A couple terms that we're going to talk about here. The term presynaptic neuron. This refers to a nerve cell that carries a nerve impulse towards the synapse. And then postsynaptic neuron. These are the cells that receive the signal and they carry the impulse away from the synapse. There are a couple of different types of synapses that we want to talk about. First is an electrical synapse. Now, at electrical synapses, action potentials conduct directly between the plasma membranes of adjacent neurons through structures we already learned about called gap junctions. Now, electrical synapses have two main advantages. They provide for faster communication, and they allow for synchronization. Next, we have a chemical synapse. At a chemical synapse, the pre- and postsynaptic cells come close to each other, but they do not touch. So that's going to be different between electrical and chemical synapses. In a chemical synapse, they are separated by a synaptic cleft, which is filled with interstitial fluid. The time required for an action potential to come to the synaptic cleft, for the neurotransmitters to cross the cleft, and for the action potential to fire in the postsynaptic neuron, this is called synaptic delay. So this delay is going to be the time it takes for everything to occur, for communication to transfer from one cell to the next. In electrical synapse, we don't have that delay because the cells are going to be in direct contact with each other. Whereas in a chemical synapse, they're not directly touching, so we have this synaptic delay. And a chemical synapse is going to be much slower than an electrical synapse. Most synapses between neurons are going to be axodendritic. So you can see here the axon connects directly to the dendrites. Others are going to be axosomatic, where the axon connects directly to the cell body or the soma. And others will be axonic, where we're going to have axon to axon connection. In addition, these types of connections can be electrical or they could be chemical. So even though we have different places that we can have synapses, the synapse could still be either an electrical synapse or a chemical synapse, just like in the previous slide. Here you can click on this link here to look at events that occur at a synapse. It's a good animation there. So here is a chemical synapse in where the action potential has reached the axon terminal in the presynaptic neuron. So this neuron at the top, this is going to be the presynaptic neuron. This is the synaptic end bulb for that presynaptic neuron. So at number 1, here we have the nerve impulse has come to the synaptic end bulb. And at number 2 here, we see that the depolarizing phase of the nerve impulse opens these voltage-gated calcium channels, which are present in the membrane of synaptic end bulbs. And because calcium ions are going to be a lot more concentrated in the extracellular fluid, the calcium rushes into the cell. You can see it here as well. So we have calcium rushes into the cell once we have that depolarization effect because of the nerve impulse reaching the synaptic end bulb. Then those calcium ions enter the presynaptic neuron. They serve as the signal that triggers these vesicles to exocytose. And these vesicles are filled with neurotransmitters. So you can see here, these vesicles will fuse with the plasma membrane, and those neurotransmitters are released into the synaptic cleft. So you can see this area here is all going to be considered the synaptic cleft. So this is the space between the presynaptic neuron and the postsynaptic neuron. So on this side we have the postsynaptic neuron, most likely the dendrites of the postsynaptic neuron. So then here at number 4, the neurotransmitters will diffuse across the synaptic cleft and bind to these neurotransmitter receptors on ligand-gated channels. So here the receptor is labeled here. You see these little openings on the ligand-gated channels. And these neurotransmitters are going to bind to those neurotransmitter receptors on the channels. And then these channels are going to open. As you can see here in number 5, we have now opened ligand-gated channels. And these allow specific ions to cross the membrane of the postsynaptic neuron. In this case is going to be sodium. So you can see sodium is crossing here into the cytoplasm of the postsynaptic neuron. Now, as the ions flow through these open channels, the voltage across the membrane changes. So this change in membrane voltage is called the postsynaptic potential. So it's going to be that change in membrane potential in the postsynaptic neuron. Now, depending on what ions the channels admit into the cell, the postsynaptic potential may be depolarization, which is going to be an excitation which is excitation causing an action potential if that depolarization reaches threshold, or it could also be hyperpolarization, which would be inhibition, which would be bringing the potential of the postsynaptic neuron further away from threshold, so inhibiting it from creating an action potential. So if sodium, which are positively charged, enter into the cell, we're going to get depolarization because we're bringing the membrane potential of the postsynaptic neuron further or closer to 0 and closer to that threshold, causing it to be less negative. If, for example, the neurotransmitters released into the synaptic cleft are, let's see, for example, chloride, which is a negative ion, we're actually going to have hyperpolarization if the chloride ions enter into the postsynaptic neuron, because it's going to bring the membrane potential further away from threshold. It's going to cause it to be more negative and hyperpolarize it. Another ion channel that can cause hyperpolarization would be potassium ion channels. Because potassium concentrations are a lot higher inside the cell than outside the cell, when we have ligand-gated potassium channels open, then potassium will actually rush out of the cell, still causing a hyperpolarization effect. So if we think about these ones with sodium here, when we have a depolarizing postsynaptic potential and it reaches threshold, it will trigger an action potential or a nerve impulse in the axon of the postsynaptic neuron. One thing to note as well, that at most chemical synapses, you can only have one-way information transfer. So you're always going to have presynaptic to postsynaptic information traveling. You're never going to have it the other way. And that's going to be at chemical synapses, not necessarily at electrical synapses. So like we just discussed in the last slide, a neurotransmitter can cause either an excitatory or an inhibitory graded potential. A neurotransmitter that causes depolarization of the postsynaptic potential, this is going to be excitatory. And this is because it brings the membrane closer to threshold. A depolarizing postsynaptic potential, this is called an excitatory postsynaptic potential. Sometimes it's just abbreviated EPSP, excitatory postsynaptic potential. Although one single EPSP normally doesn't initiate a nerve impulse, a postsynaptic cell does become more excitable if it has an EPSP. Now, a neurotransmitter that causes a hyperpolarizing postsynaptic potential, this is going to be inhibitory. During hyperpolarization, generation of an action potential is actually more difficult than usual because the membrane potential becomes more negative inside the cell, and therefore it becomes even further from threshold than at resting potential. A hyperpolarizing postsynaptic potential is termed inhibitory postsynaptic potential and abbreviated IPSP. Now, a postsynaptic neuron can receive many signals at once, and those signals can be summated to get an action potential, but as they are summated, they are still going to be considered graded potentials until they reach that threshold, and then it's considered an action potential. Each neurotransmitter receptor has one or more neurotransmitter binding sites to which a specific ion can bind to. When a neurotransmitter binds to the correct neurotransmitter receptor, the ion channel opens and you get a postsynaptic potential, either an IPSP or an EPSP, that forms in the membrane of the postsynaptic cell. Neurotransmitter receptors have two different structures. We have ionotropic and metabotropic receptors. Ionotropic receptors are types of neurotransmitter receptors that contain the neurotransmitter binding site and an ion channel. In other words, the neurotransmitter binding site and the ion channel itself are both parts of the same protein. Many excitatory neurotransmitters bind to ionotropic receptors that contain cation channels, and you're going to have EPSPs that result from opening these cation channels. And you see exactly that with this top picture. It's the ionotropic acetylcholine receptor. So the acetylcholine will bind to the binding site, the neurotransmitter binding site, which opens up this ion channel within the same protein, and it's going to allow positive ions in, causing this excitatory postsynaptic potential. We also do have the opposite ionotropic receptor here, which is a GABA receptor. So you're going to have GABA bind here, opening the ion channel, allowing calcium in, which is actually an inhibitory postsynaptic potential. So GABA is an inhibitory neurotransmitter. Now, a metabotropic receptor, this is a type of neurotransmitter receptor that contains a neurotransmitter binding site, but it lacks an ion channel as part of its own structure. However, a metabotropic receptor is coupled to a separate ion channel by a type of membrane protein called a G protein. So you have this G protein that kind of couples it with an additional ion channel. When a neurotransmitter binds to a metabotropic receptor, the G protein can either directly open or close the ion channel, or the G protein might act indirectly by activating another molecule, which is called a second messenger. Which is in the cytosol, which in turn can then open or close the ion channel itself. Second messenger is just going to be an additional protein that the G protein will activate, and then the second messenger will activate the ion channel. Some inhibitory neurotransmitters bind to metabotropic receptors that are linked to potassium channels. So you can see here we have this acetylcholine metabotropic receptor, which is going to open that potassium ion channel, and it is going to cause an inhibitory postsynaptic potential. Removal of the neurotransmitter from the synaptic cleft is actually really important for normal synaptic function. If the neurotransmitter can linger and stick around in the synaptic cleft, it would influence the postsynaptic neuron or muscle fiber or gland indefinitely. So neurotransmitters are removed in one of three ways. First one is diffusion. So some of the neurotransmitter molecules kind of just diffuse away from the synaptic cleft. They just kind of leave. And once it's out of reach of its receptors, it can no longer exert any effect. The second way would be enzymatic degradation. So certain neurotransmitters are inactivated through enzymatic degradation. An example of this would be the enzyme acetylcholine esterase. Esterase, and acetylcholine esterase is an enzyme that breaks down acetylcholine in the synaptic cleft. The third way that a neurotransmitter can be removed from the synaptic cleft is uptake by cells. So many neurotransmitters are actually actively transported back into the neuron that released them, so back into that presynaptic neuron. Other neurotransmitters are transported into neighboring neuroglia. So some of the neuroglia can actually take up neurotransmitters as well to remove them from the synaptic cleft. And that's going to be the end of Chapter 12, Part 10.